In this video, you will learn about how to attempt some MCQs on carbohydrates. Question number 1 is, when all the monosaccharides in a polysaccharide are of same type, such type of polysaccharide is called option A, disaccharide, B, homoglycan, C, heteroglycan, D, oligosaccharide. If we look into the options, option A and D, oligosaccharide and disaccharide, these are not polysaccharides. So these are wrong option. B, homoglycan, C, heteroglycan. Homo means same, hetero means different. So the right option for this question is B, homoglycan. Homo means same, glycan means saccharides. So in homoglycan are the polysaccharides in which all the saccharide units are of same type. While if we look into the option C, heteroglycan, hetero means different. Heteroglycans are also the polysaccharides, but they are made up of different saccharide units. What are the examples of the homoglycans? Homoglycan examples are cellulose, starch, glycogen, and chitin. These are polysaccharides which are made up of all the same units. These are made up of glucose units. Cellulose, starch, glycogen, and chitin, all these are made up of glucose units. Means this, all the units are the glucose. But the only difference in them, they are different from each other because the number of glucose units present in them are different. In cellulose, the number of glucose units vary from 3000 and it may be more than that. In starch, the glucose units present vary between 300 to 1000. In glycogen, the glucose units may be present between 2000 to 60,000. And in chitin, the glucose units may be present or vary between 100 to 1000. So that's why they are different from each other. Depends on the number of glucose units present in them. And the other difference is the linkages, as these monosaccharide units are linked together by glycosidic linkages. And the glycosidic linkages in cellulose is beta 1, beta 4. The glycosidic linkage forms between beta 1 and beta 4. And in starch, the glycosidic linkage is alpha 1, alpha 1 4, alpha 1 4 for the straight chain, and for the branches there is alpha 1 6 linkages. For the glycogen there is alpha 1 4 and alpha 1 6 linkages, and for the chitin there is beta 1 4 linkages. So that's how homoglycans are made up of same units, and these are the examples of these homopolysaccharides. Question number 2 is, the polysaccharide used in assessing the glomerular filtration rate is A, glycogen, B, agar, C, inulin, D, hyaluronic acid. The right option is C, inulin. As inulin are smaller in size and they cannot be metabolized in the body, so they are excreted as it is from the kidneys. So the inulin is used to assess the kidney functions. When inulin is injected to the patient intravenously, the amount of inulin that will appear in the urine over a period of time is then measured. And when the inulin is measured, the rate, it will be directly proportional to the glomerular filtration rate. Question number 3 is, the polysaccharide found in the exoskeleton of invertebrates is option A, pectin, B, chitin, C, cellulose, D, Keratin Sulfate. The right option is B, chitin. Chitin is present in the exoskeleton of invertebrates. As you can see here, these are some invertebrates given over here, and their external surfaces and external structures are made up of chitin. As they are the hard surfaces and hard structures, so the chemical composition for these structures are chitin, and chitin is a polysaccharide. Question number 4. Which of the following is a heteroglycan? A, dextrins, B, agar, C, inulin, D, chitin. If we look into the question, hetero means different, glycan means saccharides. Heteroglycans are the polysaccharides which are made up of different saccharide units. Option A, C, D, dextrins, inulin, and chitin. These are homopolysaccharides. So the right option is B, agar. Agar are the polysaccharides that are made up of different saccharide units. Agar is a heteroglycan, which means that it is a polysaccharide made up of different units of sugar molecules. So agar, it is made up of D-galactose and 3,6-N-hydro. If you look into the structure, it is made up of galactose and 3,6-N-hydro, L-galactose, and these units are then repeated again. So by these units repeating again, they make up the long chains of polysaccharides, but as they consist of different units, so they are heteroglycans. Number 5, the glycosaminoglycan which does not contain uronic acid is A. Dermatin sulfate, B. Chondroitin sulfate, C. Keratin sulfate, D. Heparin sulfate. That which among of these of the following does not contain the uronic acid? So these all are the glycosaminoglycans. If we look into these in detail, this is the heparin sulfate. It contains the N-acetyl glucosamine and glucuronic acid. Keratin sulfate, it also consists of N-acetyl glucosamine and glucuronic acid. And if we see, this is keratin sulfate. The keratin sulfate, it is made up of galactose and N-acetyl glucosamine. So it means that keratin, it does not contain the glucuronic acid. So the right option for this is keratin sulfate, as the keratin sulfate is the glycosaminoglycan, that it consists of N-acetyl glycosaminoglycan and galactose, but it does not contain the glucuronic acid, while the rest all of these glycosaminoglycans, it consists of the glucuronic acid. Other question is, keratin sulfate is found in abundance in A. heart muscle, B. liver, C. adrenal cortex, D. cornea. The right option is cornea, as cornea is the important part of the eye, and keratin sulfate is abundantly found in human body, in the most specifically in the cornea. Question number 7 is, repeating units of hyaluronic acid are A. N-acetyl glucosamine and D-glucuronic acid, B. N-acetyl galactosamine and D-glucuronic acid, C. N-acetyl glucosamine and galactose, D. N-acetyl galactosamine and L-iduronic acid. So the right option for this question is option A. Hyaluronic acid are made up of N-acetyl glucosamine and D-glucuronic acid. As you can see here in this structure, this is the hyaluronic acid single unit. So these repeating units of this unit, hyaluronic acid units, these make up the hyaluronic acid. It is made up of two different units, so it is also a heteroglycan. It is made up of D-glucuronic acid. This is the glucose unit, but if you can see here instead of CH2OH, there attached is COH. So that's why it is glucuronic acid. And if we see here, this is also a same structure of the glucose, but at this position at carbon number 2, you can see here there is amino group attached, nitrogen. So that's why it is amine. This is glucosamine, and the acetyl group CH3 is also attached, so that's why it is known as N-acetyl glucosamine. So N-acetyl glucosamine, because of this attachment of amino and acetyl group, and this is glucuronic acid because of this group, this acidic group is attached, so that's why it is glucuronic acid, and it is acetyl glucosamine. These combine together by the linkages and makes up the hyaluronic acid single unit, and the repeating units make up the long chain of the hyaluronic acid. Question number 8 is, the approximate number of branches in amylopectin is A10, B20, C40, D80. The right option is 80, as the amylopectins is one of the type of the starch that is made up of glucose units that are linked together by alpha-1,4 glycosidic bonds. As alpha-1,4 glycosidic bonds, these bonding are present in the straight chain, and then about after 20 to 30 glucose units, then after each 20 to 30 units, there is a branch point. This is the branch point in which there is the linkage alpha-1,6 glycosidic bond. So this branch chain is alpha-1,6 and the straight chain is alpha-1,4. So the question was asked the approximate number of branches. So in this amylopectin, how much branches will be attached by alpha-1,6 linkages? There are approximately 80 in number. Their question is similar, is about amylopectin. In amylopectin, the interval of glucose units of each branch is. So in this question, they have asked about the intervals. A 10 to A20, B 24 to 30, C 30 to 40, D 40 to 50. So the right option is 24 to 30, as we have already discussed in the previous MCQs that amylopectin is a type of a starch that is made up of glucose molecules that are linked by alpha-1,4 linkages, but at every 20 to 30, or it may be 24 to 30, after every 24 to 30 units there is a branch chain which is the linkage alpha-1,6 linkage. So this linkage alpha-1,6 linkage, it basically makes the branches. So from after how many glucose units the branch chain is formed, it is 24 to 30 units. The branch chain is formed by alpha-1,6 linkages. Question number 10 is, the most abundant carbohydrate found in nature is A. Starch, B. Glycogen, C. Cellulose, D. Chitin. The right option is cellulose. As cellulose is also one of the structural polysaccharide that makes up the cell walls of plants. So cellulose is present in plants, so plants makes up the entire environment. They are present very abundantly in environment. So cellulose is a carbohydrate that is found abundantly in our nature. So cellulose is not digested in humans, and it is an important source of fiber in our diet. Fiber helps to keep the digestive system healthy and can help to lower the cholesterol levels. So we take the cellulose by the plant sources, and the cellulose in human body functions as a fibers. 11. Alpha-D-glucuronic acid is present in A. Hyaluronic acid, B. Keratin sulfate, C. Heparin sulfate, D. All of these. As if we see here the glucuronic acid, this is the glucuronic acid which is present in Keratin sulfate, heparin sulfate. This is the alpha form. If we see the position of the hydroxyl ion, it is below, and if the hydroxyl position is above and hydrogen is below, then that would be beta form. But in this here, the glucuronic acid, the hydroxyl is and the hydroxyl OH is present below, so this is alpha form. The right option for this is all of these, because this alpha-D-glucuronic acid is present in all of these. In hyaluronic acid, Keratin sulfate, and heparin sulfate, the glucuronic acid is present in the form of alpha form. Alpha-D-glucuronic acid is present in hyaluronic acid, Keratin sulfate, and heparin sulfate. In all of these, it is present. 12 is, a carbohydrate commonly known as dextrose is A. Dextrin, B. D-fructose, C. D-glucose, D. Glycogen. The right option for question number 12 is D-glucose, as D-glucose, which is commonly known as dextrose. As dextrose is a type of sugar that is found in many foods and beverages, and is also available as a dietary supplement. For what purpose dextrose is used? Dextrose is a quick source of energy that can be used to treat low blood sugar level. If you see here, this dextrose, this is commonly available for intravenous solution. So dextrose intravenous solution, this is a solution of dextrose that is given intravenously and is often used to treat low blood sugar and dehydration. You have commonly seen this dextrose. So it is given to the patients intravenously to treat the low blood sugar or dehydration. 13. A heteropolysaccharide among the following is A. Inulin, B. Insulin, C. Heparin, D. Dextrin. As the name indicates that hetero means different. Polysaccharides, so heteropolysaccharides are the polysaccharides that are made up of different monosaccharide units. If you see to the classification of the polysaccharides are classified into homopolysaccharides and heteropolysaccharides. Homopolysaccharides are made up of same glucose, same units. The examples of the homopolysaccharides are starch, dextrin, glycogen, cellulose, inulin, dextrins, and chitin, while the examples of the heteroglycans are agar, gum, pectins, and glycosaminoglycans. If we look into the option, inulin comes into the homopolysaccharide, Cellulose, it is homopolysaccharide. Dextrin, it is a homopolysaccharide. So the question is hetero. So heparin, it comes into this category, glycosaminoglycans. So the right option for this question is heteropolysaccharide. Among these is the heparin. Heparin is the heteropolysaccharide. It belongs to this glycosaminoglycans. So heparins are made up of alternating units of glucosamine and glucuronic acid. So these glucosamine and glucuronic acid, these alternating units combine together to make up the long chain of polysaccharides known as heparin. Question number 14. Hyaluronic acid is found in A. Joints, B. Brain, D. Abdomen, C. Abdomen, D. Mouth. The right option is hyaluronic acid is found in joints. As hyaluronic acid is a type of glycosaminoglycan, it means that they are also heteropolysaccharides which are made up of long chain of sugar molecules. It means that they are made up of glucosamine and glucuronic acid. In the joints, what are the functions of the hyaluronic acid? Hyaluronic acid helps to lubricate the joints. They are shock absorber. They provide nutrients to cartilages, and they remove waste products from the joints. Question number 15. An L isomer of monosaccharide found in the human body is A. L-fructose, B. L-erythrose, C. L-xylose, D. L-xylulose. The right option for this question is L-xylulose. The only L isomer of monosaccharide found in the human body is L-xylulose. And this L-xylulose, they are not produced in the human body, but we take it from external sources like fruits and vegetables. This L-xylulose perform the following functions. They are involved in the health benefits in the body. They improve the blood sugar control. They are involved in the reducing the risk of heart diseases, and they also improve the gut health, and they are also involved in the protection against the antioxidants. Question number 16. Cellulose is made up of the molecules of A. Alpha glucose, B. Beta glucose, C. Both, D. None of these. As cellulose is the polysaccharide that are made up of many saccharide units joined together to form the cellulose. So which of these units are? The right option is A. Alpha glucose. They combine together by the glycosidic linkages and make up the long chain of cellulose. Question number 17 is, what is the chemical difference between cellulose and chitin? A. Replacement of the hydroxyl group at carbon 2 with an acetylated amino group. B. There is a replacement of the hydroxyl group at carbon 3 with an acetylated amino group. C. Replacement of the hydroxyl group at carbon C with an acetylated amino group. D. Replacement of hydroxyl group at carbon 5 with an acetylated amino group. If we look into the structures of the cellulose and chitin, cellulose and chitin, these both are polysaccharides, are made up of similar units. These are made up of similar units of glucose units combined together by the glycosidic linkages to form the cellulose and chitin. This is the structure of the cellulose. This is glucose unit. This is glucose unit. These make up the cellulose. And similarly, if we see to the structure of the chitin, these are also made up of the glucose unit, but this is carbon 1, this is carbon 2. At carbon number 2, there is acetylated amino acid. This is amino group attached, and CH3, there is also the acetyl group attached. So this is the glucose amine group. So this acetylated amino acid, and then again 1 and carbon number 2, there is acetylated amino group attached. So the right option is the cellulose and chitin are similar, but the difference is that this is simple glucose structure, but at the chitin, at carbon number 2, the acetylated amino group is attached. So the right option for this question is replacement of hydroxyl group at carbon 2 with an acetylated amino group. As you can see in the structure, all the structure is similar, but the difference is that at carbon number 2, there is the attachment of the acetylated amino group. Question number 18. In which of the following forms glucose is stored in plants? A. Glycogen, B. Starch, C. Dextrin, D. Cellulose. The right option is A. The storage form of glucose in plants is starch, while in animals, the storage form of glucose is glycogen. As starch is a carbohydrate that is found in plants, it is made up of glucose molecules that are linked together in chains. Starch is most abundant carbohydrate in human diet. As most abundantly in diet we take the carbohydrate is of the starch type. It is a good source of energy. What are the functions of the starch? Starch gives us energy. They are used as a fiber. They also give us satiety. It also helps to promote the heart health. Question number 19. In which of the following forms glucose is stored in the liver? A. Glycogen, B. Starch, C. Dextrin, D. Cellulose. The right option is A. Glycogen. Glycogen is the storage form of glucose in the animals, or also you can say in the human body. The glucose is also stored in the form of the glycogen. Whenever we need glucose, it is utilized for the energy, and when there is enough energy, then glucose they combine together by glycosidic linkages and they makes up the glycogen, and glycogen are stored in the liver. As you can see the structure of the glycogen, it has the straight chain alpha-1,4 linkages and the branch chains alpha-1,6 glycosidic linkages. So glycogen is a polysaccharide that is stored in the liver and muscles. It is made up of glucose molecules that are linked together in branch chains. Glycogen is the main source of energy, and whenever we need energy, it is broken down into glucose and it fulfills the needs of the body energy. Question number 20 is, which of the following are structural polysaccharides? A. Glycogen, B. Starch, C. Chitin, D. Glucose. If we look into the options, glycogen, starch, these are the polysaccharides that are the storage form of polysaccharides, and glucose, it is the quickest source of energy. So the left option is C. Chitin. So chitin are the polysaccharides that are made up of repeating units of N-glucosamine, and it is the second most abundant biopolymer in nature after cellulose. So it can also be used as a biopolymer. Chitin is found in the exoskeleton of insects, the shell of crabs and lobsters, and the cell walls of the fungi. It is also used in a variety of applications they are used for different purposes. For food additives, they are used in cosmetics, and they are also utilized for pharmaceutical purposes. Question number 21 is, starch consists of A. Branched amylose and branched amylopectin, B. Unbranched amylose and branched amylopectin, C. Unbranched amylose and unbranched amylopectin, D. None of the above. So if we look into the structure of the starch, this is the amylose. Amylose is the straight chain. It is unbranched, alpha-1,4 linkages. So this is unbranched chain in starch is known as amylose, while the branch chain, which is linked by alpha-1,6 linkage, it is known as the branch chain in starch is known as amylopectin. So the right option for this question is, starch consists of unbranched amylose and branched amylopectin. This is the unbranched amylose, which is joined together by alpha-1,4 linkages, and the branch chain is known as amylopectin, which is linked by alpha-1,6 linkages. Question number 22. Oligosaccharides linked to proteins are called A. Glycolipids, B. Glycoproteins, C. Galactosides, D. Ganglioside. The right option is, when the oligosaccharides link to the protein, they are known as glycoproteins. If we look into the structure, this is the structure of the monosaccharide, but as oligosaccharide, when they are linked to the proteins, this is the amino group, so they are known as glycoproteins. Glycose for the saccharides and the proteins is for the proteins or amino groups, so they makes up the glycoproteins. Glycoproteins perform various functions in the body. Glycoproteins are the types of carbohydrate protein compound that contain carbohydrate molecule that are covalently bound to a protein. Glycoproteins are found in all living organisms, and they have a variety of roles. What functions are performed by the glycoproteins? They are involved in cell signaling, they are involved in immunity, they have a role in cell adhesion, they are involved in transport, and they also contribute to the structure of the cells and tissues. Question number 23. The complete hydrolysis of amylopectin yields that if the amylopectin is hydrolyzed or broken down, it will give rise to A. amylose, B. galactose, C. glucose, D. fructose. The right option is, as amylopectin, they are made up of glucose units. So when the amylopectin is hydrolyzed, they will give rise to glucose. If you look into the structure, this is the amylopectin, which is the branch chain, and these amylopectins are made up of the glucose units. So when amylopectin is hydrolyzed, it will give rise to glucose units. Amylopectin is a type of starch that is made up of glucose molecules that are linked together in a branch chain. It is most abundant form of starch in plants, making about 80% of starch molecules. So the most abundantly that is present, the plant source that are taken up by humans as a diet source, they are 80%. Amylopectin is found in which food they are found? They are found in seeds, they are found in roots and tuber of the plants. Amylopectin is a homopolysaccharide, which means that it is made up of only one type of molecule, which is glucose. So all the units are glucose, and when the amylopectins are hydrolyzed, it will give rise to the many glucose units. Question number 24. Which of the following is true about inulin? A. Is present in muscles. B. Is a homopolysaccharide. C. Decreases blood glucose. D. Is metabolized in liver. The right option is inulin is a homopolysaccharide. Inulin is fructane, as inulin is a homopolysaccharide which is made up of same units combined together, and these units are the fructose. So inulin are made up of same fructose units linked together by the glycosidic linkages and makes up the polysaccharide known as inulin. Question number 24. Cellulose, a complex carbohydrate or polysaccharide consisting of dashed glucose units. Like how many glucose units are present in cellulose? A. 100 to 1000, B. 1000 to 2000, C. 3000 or more, D. 50,000 to 60,000. The right option is, as we have already discussed about the different number of glucose units present in the homopolysaccharides. So the right option for the cellulose is C. It consists of 3000 or more than that similar glucose units combined together by glycosidic linkages and makes up the complex structure of homopolysaccharide known as cellulose. And cellulose is the structural polysaccharide that make up the cell wall of the plants, and that are taken up by the human body as a dietary source and which function as a fiber in a human body. In this video, some of the MCQs were explained. If you want to learn more, keep watching. Thank you.